Next up is Andy Stone. Andy is a business development officer with the NRC here in Halifax, and over the last while has been seconded on a part-time basis to Capital Health, and served in that capacity as part of that secondment as a project coordinator to help implement a mutational screening technology for lung cancer in Atlantic Canada. And I would bet that every single one of us in this room has been impacted by lung cancer, either directly or indirectly, and hence very interested to hear what Andy has to say. He's representing the dedicated efforts of a number of folks that have been involved in the project, so I'll let Andy offer the thank yous to them. And Andy, the floor is all yours.
Thanks, Steve. So I'm going to talk about a project that's happening literally down the street, the Mackenzie building. And it's specifically lung cancer, and I think it's in our vision that it's just the first of many different types of cancer. And what we're doing is enabling, we're kind of what I would call the back end of a lot of genomics research. In fact, I think we're actually bookended by genomics. The state that we're at right now, especially with lung cancer, is not a particularly pleasant one, where the success rates with a lot of the chemotherapies that are available today are between 15 and 20%. And what's happened in the last few years, though, is there's been some very promising new targeted therapies that completely are changing the game and increasing the response rates up to 60 to 75%. And furthermore, with what we're looking at doing is actually what was missing is this ability to what's called stratify the patient population and pull out the people who are actually have the mutation that can be targeted for. So what was missing was really a means of identifying, which patients have which mutations. And when you do this type of process, what we find is that actually the average goes up for pretty much all groups within this process, so that it's known now that giving a targeted therapy to somebody that doesn't have the mutation is in some cases worse than doing nothing. So by this whole process, it really enables the rational application of chemotherapies in the healthcare system. So it's definitely going to be better for the patient outcome, but we also think with a rational drug delivery approach will actually show benefits within the healthcare system itself. So when we break down lung cancer, we see that a little more than half of lung cancer patients have a known mutation. And what we've done at Capital District Health Authority with the help of NRC is to develop what we call a multiplex panel, or the ability to test for multiple mutations in one screening. So when the patient has a biopsy taken, part of the process now will include not just the typical pathology reports on, that categorize the type of cancer and the stage of cancer, but also now molecular profiling. So we're going to see, this is the front end of a game changer within this space, that patients will not only have kind of the traditional anatomical and stage of your cancer included in your diagnosis, but also kind of a molecular signature which is going to have an impact on your treatment pathways, as I suggested. So what we have done is taken at CDHA the clinically actionable mutations, put those on our screening panel, and have just implemented this in a pilot phase right now, starting in September. I think we have about 55 patients that have actually gone through this pilot clinical program, and of those, quite a few have shown a targetable mutation, and instead of getting the traditional first-line therapy, we'll now immediately get the targeted therapy. So we're already having an impact on patients' lives. This project has happened quickly, and it's been, for me, it's actually been surprising when people come up to me from Toronto, Vancouver, and even some of the pharmaceutical companies and ask, well, how did you get this done here? And it really comes down to the team that's on the ground, and fundamentally has to be multidisciplinary. This includes surgery, medical oncology, pathology, the lab, as well as a lot of other folks behind the scenes. And the implementation of this technology within the hospital system is actually proven to be much more complicated than the technology itself. But it's the dedication of a team like this. And I won't read off everybody's names. We'll leave this up for a moment. And this also has been funded through a number of different pharmaceutical companies. And what's really interesting is for a consortium like this to work is that when you look at the different types of mutations, they tend to be mutually exclusive. So the pharmaceutical companies aren't really competing in this particular instance. You either have mutation that would lead you towards their drug or you don't. So they're all happy to develop kind of this multiplex screening technology so as a mechanism to direct the patient towards whatever drug is most appropriate for them. And then just to finish off on why I think this is bookended by genomics applications is that the technology platforms we're employing right now really only allow us to look at a relatively small number of mutations, and we know already, we're already thinking of how are we going to supersede ourselves, and it's probably with a genomics platform probably honed in specifically on the cancer exome, so just those genes that are involved with cancer, which would reduce the amount of data by probably to 1/1000th. And looking to now screen in the future all patients for their entire cancer exome and building a tremendous amount of data that can be mined retrospectively in a clinically annotated database. So it's quite a future, I think, ahead of this group. So, great, thank you.